Imagine! Regeneration is an organism's ability to recreate lost or damaged tissues, organs, and limbs. Since ancient times, people have been fascinated with the idea of regeneration. In Greek mythology, the Hydra was a monster that could grow back 2 heads for every one cut off. In Chinese folklore, there is a fox with 9 tails, each tail corresponding to a life she could lose on her quest to become human. Every time she died, she would lose a tail, but could regenerate a living body from just one of the remaining tails. While you'd have trouble finding such exotic examples of regeneration today, the phenomenon still exists to varying degrees within different organisms. For example, salamanders and newts have the ability to regenerate tails, limbs, jaws, eyes, and various internal structures. Planaria, or flatworms, are more remarkable still since they can regenerate a complete organism from even a small fraction of their original body. Echinoderms, such as the starfish that we study in our lab, can also regenerate. The starfish, if threatened by a predator, can detach and sacrifice its arm in order to get away, and then regenerate a new one later. Some starfish can even regenerate from a single arm and portion of its center. Even humans have the ability to regenerate, though we are much more limited. We have the ability to heal wounds when we get a cut or break a bone, and recently there have been a number of cases of people who were able to completely regenerate the tip of their finger.
Our project examines regeneration in larval starfish. Starfish larvae were cut in half along the red line pictured to produce head and body portions. The regeneration process was then observed under a microscope. At various time points in the process of regeneration, we examine the expression of a gene called GCM that plays an essential role in neuronal development. In this image, dark purple spots mark where GCM was turned on 3 days after beginning regeneration. Why should we care about a regenerating starfish's gene expression? By investigating the gene expression involved in regeneration, we can get a better understanding of how that trait evolved over time between different species, and extend our knowledge to that of humans. With this knowledge, we can develop new technologies in regenerative medicine, which encompasses a diversity of applications, from organ transplants to amputations. Currently, it's estimated that approximately 1 in 3 Americans may benefit from regenerative medicine. Understanding regeneration in other organisms is complementary to understanding new possibilities in human health sciences.